Schwann cells or neurolemmocytes are the principal glia of the peripheral nervous system. Glial cells function to support neurons and, in the PNS, also include satellite cells, olfactory ensheathing cells, enteric glia and glia that reside at sensory nerve endings, such as the Pacinian corpuscle. There are two types of Schwann cell, myelinating and nonmyelinating. Myelinating Schwann cells wrap around axons of motor and sensory neurons to form the myelin sheath. Schwann cells are involved in many important aspects of peripheral nerve biology, the conduction of nervous impulses along axons, nerve development and regeneration, trophic support for neurons, production of the nerve extracellular matrix, modulation of neuromuscular synaptic activity, and presentation of antigens to T-lymphocytes. Charcot-Marie tooth disease, Guillain-Barré copyright syndrome, schwannomatosis, acute inflammatory demyelinating polyradiculopathy, and chronic inflammatory demyelinating polyneuropathy, and leprosy are all neuropathies involving Schwann cells. Description: named after the German physiologist Theodor Schwann, Schwann cells are a variety of glial cell that keep peripheral nerve fibers alive. In myelinated axons, Schwann cells form the myelin sheath. The sheath is not continuous. Individual myelinating Schwann cells cover about 100 micrometers of an axon, equating to approximately 10,000 Schwann cells along a 1 meter length of the axon, which can be up to a meter or more in length. The gaps between adjacent Schwann cells are called nodes of Ranvier. The vertebrate nervous system relies on the myelin sheath for insulation and as a method of decreasing membrane capacitance in the axon. The action potential jumps from node to node, in a process called saltatory conduction, which can increase conduction velocity up to 10 times, without an increase in axonal diameter. In this sense, Schwann cells are the peripheral nervous system's analogues of the central nervous system's oligodendrocytes. However, unlike oligodendrocytes, each myelinating Schwann cell provides insulation to only one axon. This arrangement permits saltatory conduction of action potentials with repropagation at the nodes of Ranvier. In this way, myelination greatly increases speed of conduction and saves energy. Non-myelinating Schwann cells are involved in maintenance of axons and are crucial for neuronal survival. Some group around smaller axons and form Remak bundles. Myelinating Schwann cells begin to form the myelin sheath in mammals during fetal development and work by spiraling around the axon, sometimes with as many as 100 revolutions. A well-developed Schwann cell is shaped like a rolled-up sheet of paper, with layers of myelin in between each coil. The inner layers of the wrapping, which are predominantly membrane material, form the myelin sheath while the outermost layer of nucleated cytoplasm forms the neurolemma. Only a small volume of residual cytoplasm communicates the inner from the outer layers. This is seen histologically as the Schmidt-Lantermann incisure. Schwann cell transplantation and regeneration. A number of experimental studies since 2001 have implanted Schwann cells in an attempt to induce remyelination in multiple sclerosis-afflicted patients. In the past 2 decades, many studies have demonstrated positive results and potential for Schwann cell transplantation as a therapy for spinal cord injury, both in aiding regrowth and myelination of damaged CNS axons. Schwann cell transplants in combination with other therapies such as chondroitinase ABC have also been shown to be effective in functional recovery from spinal cord injury. Indeed, Schwann cells are known for their roles in supporting nerve regeneration. Nerves in the PNS consist of many axons myelinated by Schwann cells. If damage occurs to a nerve, the Schwann cells will aid in digestion of its axons. Following this process, the Schwann cells can guide regeneration by forming a type of tunnel that leads toward the target neurons. The stump of the damaged axon is able to sprout, and those sprouts that grow through the Schwann cell do so at the rate of approximately 1 mm a day in good conditions. The rate of regeneration decreases with time. Successful axons can therefore reconnect with the muscles or organs they previously controlled with the help of Schwann cells. However, specificity is not maintained and errors are frequent, especially when long distances are involved. Because of their ability to impact regeneration of axons, Schwann cells have been connected to preferential motor reinnervation as well. If Schwann cells are prevented from associating with axons, the axons die. Regenerating axons will not reach any target unless Schwann cells are there to support them and guide them. They have been shown to be in advance of the growth cones. Schwann cells are essential for the maintenance of healthy axons. They produce a variety of factors, including neurotrophins, and also transfer essential molecules across to axons. Ganglioside, 9-O-acetyl-GD3 ganglioside is an acetylated glycolipid which is found in the cell membranes of many types of vertebrate cells. During peripheral nerve regeneration, 9-O-acetyl-GD3 is expressed by Schwann cells. See also, electrophysiology, Hodgkin-Huxley model, neurotransmission, olfactory ensheathing cells, Schwannoma, references. External links, diagram at clc.uc.edu, histology image, 21301 low, histology learning system at Boston on Ivosicia ultrastructure of the cell, myelinated axon and Schwann cell, cell-centered database, Schwann cell.